Well, good evening, everybody. Welcome to Nature Live here in the Attenborough Studio, and welcome to European Researchers Night. It's great to see so many of you joining us tonight to find out more about some of the incredible research that's going on both here at the museum but also across Europe. This is our annual Festival of Science, so it's great to see you all here today. Just out of curiosity, hands up if this is your first time coming along to one of our Nature Live talks. Quite a few first-timers. Excellent. Well, let me tell you very quickly just a little bit about what Nature Live is all about. Now, when you're wandering around the museum, even if you go around all of the galleries and all of the exhibitions, which can take quite a while, you actually would only be seeing a very, very small part of what this museum is all about. And tonight is a really special night because we bring out not just specimens, but also the people that work with that incredible collection. And no doubt you've seen already, as you've been walking through the galleries, some of our amazing teams of researchers that work with our collection here every day. And Nature Live is a program where we invite speakers from here in the museum and beyond to actually come and talk to you about their work. But more importantly, it's a chance for you guys to find out and ask questions about what research is going on out there. And tonight is absolutely no exception. And I'm delighted to introduce you to a special speaker this evening. This is Mike Bruford from Cardiff University, and he's going to be talking to us about a fascinating area of conservation genetics. So if you've got any questions during the show, please pop your hand up. We've got microphones. So if you've got a question, we'll bring a microphone over to you, and you can ask your question directly to Mike. We've also got a very special treat for you as well, because we're going to be linking live to our molecular collections facility here in the museum. And that's a part of the museum that is never open to the public. You never would usually get to see this, but we're going to link there live to my colleague Camilla, who's going to be waiting for us in a few minutes. So we'll look forward to that. It's fascinating to see what goes on behind the scenes. But before we do that, thanks very much, Mike, for coming along today.
Pleasure.
Before we get started, can you sort of introduce yourself and tell us a little bit, very briefly, about what you do, what your sort of specialism is?
So I work at Cardiff University, and I'm a biodiversity scientist. What I'm mainly interested is the planet's genetic diversity. And what I also do is I'm coordinating the establishment of the UK's first zoological biobank that spans right across all of the institutions that we have. So from the Natural History Museum here, you'll see that in a minute, but also zoos, university laboratories. And so we're trying to bring together all of the material that we've collected over the last many decades, actually, and use that to build a biobank that can be used for future studies and research.
Fantastic. So we'll find out in a little bit exactly what biobanks are. But the interesting thing, you're a sort of conservationist, but you're dealing with not so much the kind of the whole animal. It's the genetic material, which I think is an aspect of collections that a lot of people might not realise. You know, when we think of a museum, we think of skeletons and things like that.
Yeah.
Is it quite a sort of futuristic area of conservation?
To some extent, although we've been studying genes now since the 1950s, and we know that genetic diversity, the variation that explains the difference between almost all of us in this room, but also between species, is the raw material that we need for species to adapt and survive. And we're living in an era of unprecedented change, climate change, land use change, everything. And without that genetic diversity, we know that biodiversity won't survive into the future. So actually genetics becomes more important the more alterations that we make to the planet and the more stress that we put biodiversity under.
Well, that's something that when we were creating Researchers Night, we were looking at, what could we call it? What would be sort of the big issues we deal with? And we came up with this idea of Planet 2.0 and sort of recognizing that scientists here understand that the Earth is in a precarious position right now, and a lot of the research, like the work that you're doing, is actually looking at what is our future going to be like and how can we live on our planet in a more sustainable and positive way.
Yeah.
All right, well, we're going to go live now over to our molecular collections facility just to see what another side of the museum collection is like. As I say, this is a museum collection that perhaps you haven't really considered before. So we're going to head over there now. Hi there, Camilla. Can you hear me?
Hey, Alistair, and hi to everyone in the studio. Thanks for joining us this evening. I'm in a behind-the-scenes location here at the museum, which is a little bit special, and I guess the likelihood is that most people in the audience won't have seen this facility, so I'm really excited to show you. And I'm joined by one of our museum scientists, Aidan Emery. Aidan, tell us a bit about what you do here at the museum and where we are.
Well, I'm one of the research staff here at the museum, and the thing that links a lot of my projects together is that I work on DNA sequences, you know, comparing different species, things like that. So we need to store the samples for doing that kind of work in you know, outside of the usual museum collection. And the best way to store DNA is to freeze the sample.
Excellent. Okay, so we can see these freezers over here, and it's chilly in here. I can feel that. Yeah. How many of these freezer units do we have in the museum?
So here in the molecular collections facility here, we've got 17 of these -80 freezers. That's minus 80 degrees Celsius, and then we've got 24 -20. So minus 80 is what you'd call ultra cold. Minus 20 is the same as your domestic freezer at home.
It all sounds pretty cold to me.
Yeah.
So it's quite a lot of freezers. How many specimens is that?
Well, it depends how big the specimen is. I mean, we've got all sorts of different shapes and sizes of container that we keep them in, so these are reasonably small. But of this size, we could get close to 2 million. We can get smaller than that, we could double that, or obviously, but they all fit in that same sort of footprint, but they're different heights. So that's how it changes.
And so those are obviously very small. Do we have any bigger, any sort of whole animals within the freezers here?
Well, we tend not to keep whole animals down here unless they are very small. So. I think the mammal section has got you know, a bigger freezer for keeping large things in. Yeah, but we tend to take, because we're interested in the DNA rather than the whole sample. Usually, it's not usually necessary for us to keep the whole thing.
Sure. Okay. Yeah. So I wonder if we can take a closer look at one of the specimens. Would you be happy to open?
Yeah, sure. So I've got some dry ice here. So the dry ice is about the same temperature as the freezer, -80. So I just grab that because I don't want the sample to thaw out. So if I grab that. So if you want to have a quick look there, you can see how it's all arranged. It's a little bit frosty. That's the problem, of course, with freezing anything, that you get condensation, and then that freezes. But there we are. If I could put it down there. So then, if we have a look at the box, so you can see the samples are all arrayed there in these little tubes. This is 48 tubes in the box here. Now I think I'm probably going to have to take that off to do this. So if I get this sample out here, I don't know if you
can we get a look at that?
Can you see that?
Be able to get with the camera.
This is an insect specimen. It's a bumblebee, and you can see this is a whole animal. Don't know if you can see that.
So it frosts up very quickly.
It frosts up very quickly. The easiest thing actually is to give it a wipe with a little bit of ethanol like that. But I better put it back down there so it doesn't cool off. So if you want to know more information about that, we keep all the information in a database, and there's nothing written on the tube because it's quite difficult to see on this side of the container. But you can see there's a barcode on the bottom of the tube.
Great. So this is quite different to it, because a lot of the specimens in our collections, entomology, for example, our insects are pinned with data labels with all the information about location, when it was collected.
Absolutely.
So show us how all that data is stored with the frozen specimens.
So if this is a little barcode reader here. So if I put that on there, hopefully you can see the barcode has been read into the tube.
Yes.
Can you see that?
Yes.
So then if I do a search on the database there, then that'll bring the specimen up. If I look at that in more detail, then that's the whole box there. You can see it's laid out in exactly the same way. That's the sample that we pulled out, and you can see it says it's. You'd probably need your reading glasses on to see it, but there you go, Bombus monticola. Now that is that's a kind of bumblebee. It's called the bilberry bumblebee. It's a Scottish thing. Now if we look at that little bit of the data there, that links to our public data portal. So if I copied that into a browser, then it brings up the specimen record here, and you can see that we've got all the details about who collected it, where it was collected from, and when. So it was collected in 2013 from Coinafern in Scotland. That's near Inverness.
Excellent. Okay.
So,
and that particular specimen that you showed us is part of a project, right, about pollinators?
yes, that's right. So it was a project that went on over several years, and they did their immediate analysis for it, and then so now it's been archived here. But there's a great deal of extra information that we could find. I mean, it is the whole the whole bee with everything, the pollen that's associated with it, the parasites that's in the pollen, and of course information from the DNA sequence of the bee itself, which is all locked up in that specimen.
So all that kind of molecular DNA information will be locked away for potentially years to come.
Yep.
And I guess in terms of food security,
Yep.
that just thinking about the broader context, that could be really valuable for us to think about the state of pollinators today compared to years ago.
Absolutely.
Excellent.
Compare with what we've got now, or what we had in 2013, with what we've got in subsequent years.
Fantastic.
Yeah.
Well, thank you so much.
Okay.
We'll hand over back to you in the studio, Alistair, but we will see you again very shortly.
Excellent. Thank you very much, Camilla. It's great to see in that location. I've only been in it a couple of times myself.
Yeah, I think Aidan is very modest, but whilst there are frozen facilities all over the UK, both in zoos and in universities and in other museums, this is probably the biggest. That whole barcoding, sample storage and preparation, and databasing is probably the gold standard really for what we need to be doing into the future.
So are storage facilities like these, are they primarily used for organisms that are at risk of extinction? Is that their primary aim, or is it a case of we want to preserve the genetic material of everything?
They're used for both, and the reason for that is that the things that we have that we believe to be common or not declining now may well become declining in the future. So in the case of the bumblebee that you just heard about, that sample was collected in 2013. Who knows what the population will look like in 20 or 30 years' time? And we can't be complacent. So these collections are actually storing material from both common and rare species, and actually the evidence that's accumulating at the moment says that even common species across the globe are losing their genetic diversity, because whilst they're not numerically small yet, they're being fragmented because of land use change, and they're becoming increasingly stressed by things like climate change. So we're seeing changes in genetic diversity even in common species.
And what are the main drivers of the extinction? You've talked a bit about climate change, which I think is one that everyone's familiar with. Is that the primary thing that's leading us to kind of we need to address this now? Is extinction at a higher rate than it's ever been before? And is it
is,
caused by
as far as we know, it's very difficult to measure extinction rates because the comparisons that we have today, which are our records that we've kept as people, as humans, are very difficult to compare with the fossil record, because that's over much longer periods of time. But yes, extinction rates seem to be occurring much, much more rapidly than previously. Perhaps the most compelling statistic is some work that was done by the Living Planet Index, which basically says that since 1970, we've lost about 50% of our larger organisms. We don't know really about the invertebrates like the bumblebee, but for our larger animals, we've lost about 50% of them.
I mean, that's an incredible figure, really, when you start to think about it. And we are to blame for that largely. It's pollution, overfishing, bad use of land.
So climate change is a secondary effect of human population growth and industrialization. And so fundamentally, it is the way that we've used and are using the planet's resources that has resulted in what we find now.
So how do you go about prioritizing what is preserved in these frozen archives?
Yeah.
Is it driven by what is close to extinction, or is there a bit more nuance to it than that?
So I think that's a very interesting question. And one of the things that we're beginning to understand when we look at these collections is that the representation of different species is not even. So as you might imagine, some of the charismatic species, they're very heavily represented in frozen collections because people are interested in them. They go out and collect them, just like the giant panda, for example, which I've worked on for quite a few years. But if we talk about the the bilberry bumblebee, they're far less well represented. So we have to look at both the representation of all of life on Earth to make sure that we're retaining the genetic material from as wide a representation of evolutionary history as we can. But then we also have to be cognizant of the fact that there are many species that are really important as indicators, as sentinels of environmental change. And the pollinator initiative is extremely interesting in that context, because pollinators are so important because they provide us with ecosystem services. They pollinate. Without pollination, all of our crop agricultural activities will founder. So it's not simply about representing all of life on Earth, but we also have to be cognizant of what's important for the future generations.
Yeah, that point you make about pollinators is particularly interesting. And in fact, we've got a whole zone as part of research tonight called Food for Thought, which is exploring our relationship to food, our plants, but the pollinators is a part of that. Yeah. Because if the pollinators go, then many of these plants that we rely on will go as well. Yeah. Now, if that were to happen, if a pollinator, let's say a pollinator were to disappear through a catastrophic change in the environment or some other human effect, could we go into these banks of genetic material and bring that species back? Would that be possible?
You're talking about de-extinction. That's a question I get asked a lot. And the simple fact is that we can't at the moment. And there's a lot of reasons to believe that we shouldn't be doing that either, because even if, under some future scenario where we could recreate organisms a la Jurassic Park, just because we could reassemble the carbon atoms in the right order and perhaps make something that looks like a brontosaurus, doesn't mean to say it's going to behave like one, doesn't mean to say it's going to engineer the landscape like the brontosauruses did 80 million years ago. So what people are tending to do now is use what we call ecological proxies. So for example, if a giant tortoise goes extinct on one particular island, let's say in the Indian Ocean, you could potentially bring in a similar species from another island and see if it is doing the same thing, if it's performing the same services. And if that's the case, then you can use that as a proxy and manage the landscape and manage the biodiversity accordingly. However, the important thing about genetic diversity is that if you're going to do that, you're going to move populations around, you better understand their genetics and how they're going to adapt to their new environments. That's where the DNA comes in.
I was about to say, because there's quite a few examples of animals that have been introduced into new areas and have caused catastrophic problems. I mean, the textbook example that comes to my mind is the cane toad, which I think a lot of people might have learnt from in school. Introduced and had these unforeseen effects. So that must be something you're very
accidental introductions or even ill-advised deliberate introductions have been responsible for more extinctions than any other kind of activity in the last 50 years. So we know that that's a real problem. What I'm talking about, though, is a smarter way to actually manage biodiversity in the landscape, acknowledging the fact that we are missing some of these keystone species and under controlled conditions, reintroduce them. Of course, it's highly controversial. People are talking about reintroducing the Eurasian lynx into the UK landscape, mainly because of browsing damage by medium-sized deer in woodlands. What will be the knock-on effects? People have reintroduced the beaver into the landscape in different parts of the UK. All the evidence is that the beavers are having a fantastic effect in water retention and stopping flooding during extreme climatic events because of the way they build their dams and because of the way that water is managed in the catchments where they exist. So there can be beneficial outcomes for sure, but there may also be unforeseen consequences.
So you mentioned earlier, you talked about the sort of ecosystem services and how organisms are providing that service almost. Is that what you're sort of, when you refer to the fact that the beavers in this instance have that positive effect on water retention in the landscape? And is that kind of what you mean when you talk about that service?
Yes, I think it's important that we don't just view biodiversity as utilitarian, as something that provides benefits to us, but if we can have a win-win situation where we both increase environmental control and increase biodiversity at the same time, I think that's an important potential future sort of smart way of working our way out of this biodiversity crisis.
Excellent, thank you. Well, I've been asking a load of questions here, but do you guys have any questions you'd like to ask Mike about this work in conservation and genetics? Yes, I'm just bring the mic over for you.
Hello, hi. I was wondering, how far does the collection go back in terms of what genetic data you have available? So that was from 2013, the bee. Do you find you've got specimens going back to, say, the 70s or the 60s at all?
Much further back than that. The vast majority of the material, and I don't work here, so I'm just making this up, okay? But I have sampled many specimens from this very museum. A lot of that goes back to the Victorian collecting eras. And of course, we can go even much further back than that. So it's now possible to extract DNA and sequence the genomes of individuals from their bones that may be tens of thousands of years old. So we have now genome sequences for example, for Neanderthals and Denisovan humans, and those are, up to 30,000 years old. It's very difficult, it's challenging to do. But it is certainly possible to get DNA sequences for very, very ancient material indeed. And in fact, the kind of material that Aidan was just showing there, which is relatively recent, the most important thing for us is we know the DNA is in good condition when we get it, but having the best possible approaches to make sure that it retains its intactness into the future. That's something that we're really looking very hard at now, because we're not just talking about something that's going to be available in 50 years' time. This is Futurama. We want it to be available in thousands of years of time as well.
Excellent. Thank you very much for your question. And an interesting point there. Yeah, we want this stuff to last for a very, very long time. Now, we saw on the first link there some of our freezers that are storing things at temperatures of about -80. But we're going to head back over to the Molecular Collections facility now to look at this place where we keep stuff at extremely low temperatures. Over to you, Camilla. Where are you just now?
Behind the scenes space, just next to the room that we were in with all the freezers. As you can see, it looks a little bit different, not drastically different, but a little bit different. And I'm joined by Jackie and Kirsty. So Jackie, tell us a bit about what you do here and what this facility is all about.
I'm Jackie-Mackenzie Dodds, and I'm manager of the Molecular Collections facility as a whole. This is my favourite room. This is where it's the coldest that it's going to be ever down here. These are liquid nitrogen tanks, so they're holding the same sorts of material as Aidan was talking about earlier. DNAs and tissues, but really, really cold. So at -196 degrees C in these liquid nitrogen tanks.
So would you say this is the coolest part of the museum?
It's the coolest part of the museum.
That was awful, but I did it anyway. Okay, great. And Kirsty's helping on particular projects.
Kirsty is just started on the Arks project with me and Mike and Aidan. And yes, we're all starting to gather together zoos, aquaria, those kinds of aggregated collections to put a lot of it into these tanks as well.
Excellent. Okay, so liquid nitrogen in these tanks, and for those of us who have, I won't tell you how many years it's been since I've studied chemistry. What exactly is liquid nitrogen? How's it so cold?
Right, liquid nitrogen. Nitrogen is there's a lot of nitrogen in the air that we breathe normally. 70% of the air mix is nitrogen. If you can, there's oxygen, there's carbon dioxide, there's other things in the air. You can liquefy the air and separate out those different gases, so you can separate liquid nitrogen out, but it has to be extremely cold to do that.
So extremely cold, as you might have said already. How cold?
-196 degrees Celsius. Yes.
Chilly.
And I have some if you want to see
I
it.
was going to say, can we take a closer look at some?
Right. Well, I have to put this visor on.
Great stuff. Good.
It's prepared for you in here.
Okay, I will step out the way actually.
A little bit of fuming here. That's the frosting of the humidity in the air coming into contact with that nitrogen. Now, what I'm going to do is try and pour it into this little container so that you can see the actual liquid nitrogen. It's a little bit of there we go.
Ooh, okay.
So there it is. Can you see that pouring? This is actually liquid nitrogen. There we are.
Wow, and just gently overflowing there.
Okay. And we can show you what it does.
Yes, yes, please do.
And I'm going to get Kirsty to help me here.
Right, so
We
tell
have
us what you're
a
about to do.
beautiful sacrificial rose
That's
here.
very romantic, that's lovely.
Yes, so, you know, this is the classic one. We're going to dip this in, and this is nice and floppy and smells nice, and it's really wonderful. I'm going to hold it in here for a bit, and then we're going to smash it to show you how brittle it becomes and how solid it becomes.
Great, I love a smashed rose.
Ooh.
Wow. So how long would you need to hold that under there for to make that?
It's solid
not that
now,
long.
so go
Wow.
for it, Kirsty.
It's like the anti-Valentine's,
Isn't that
isn't it?
amazing? So it's a
incredible.
very good way to make potpourri if you want to, because these when this dries up, it's all nice little fragments for you to put in your
You've got some
potpourri
liquid
bowls.
nitrogen lying around, why would you not make your own potpourri?
And it actually still smells quite nice too.
Yeah.
Like that.
Excellent.
Okay, so
Thank you.
yes, that's kind of what it does. So it is extremely cold, and so you'd put your biological samples in there, and they'd freeze in exactly the same way, and that prevents any degradation happening for hundreds, maybe thousands of years.
Amazing. Okay, so would it be at all possible? I was going to say I will definitely be the person who kicks that over. So would
Yeah,
it
I'll
be
put this-
possible to maybe open up one of these?
Yep,
tanks
sure,
and look
sure,
inside?
yes.
I'm going to stay out of the way again.
Again, Kirsty, do you want to open up this? This tank is it's called Walrus, and
It's not actually walrus inside the
no,
tank,
it's
okay,
its nickname, and it's a famous tank because it's been on television and it's had some of David Attenborough's samples put in it, which we're going to show you. David Attenborough was filmed putting some samples in here for the Frozen Ark project, which I think Mike is telling you about the different sort of Ark collections. So when you open this up, it's not full of liquid nitrogen, it's actually nitrogen vapour, which is why that's minus 178, not minus 196. But it's much better for us to be able to handle things in there. You can't see anything though, so I'm going to defog this by just
what's
pressing that
actually
button.
doing,
That is pulling nitrogen from a big tank behind into the bottom and pushing out the nitrogen at the top. And
okay.
this is our favourite bit because it's so beautiful when this
It is
happens.
magical.
And you can feel it's quite cold.
Oh, and you can start to see inside a bit better now. I
You
see.
can start
Yeah,
to see
yeah.
inside. So if Kirsty, can you see? Okay.
Wow. Oh, I can feel that. It's so cold.
So this is the rack. This is our favourite rack, and these samples at the top are the samples that David Attenborough put in for the Frozen Ark collection, and that is Indian elephant blood. She was wild caught, the Indian elephant, and a bit of a celebrity creature herself. You can look up about her. I can tell people later, but that's Frozen Ark endangered animals from Frozen Ark collection. The rest of these samples are some of my samples from Vietnam, and they're actually invertebrates. They're snails from endangered parts of Vietnam.
And they're stored as similarly, like what Aidan was showing us before. They're just little vials.
Yes. So we won't get one out now, but they look
That's
exactly the same, and
You've
they've got the
already smashed the
barcodes
rose
at the bottom.
first.
No, we don't want to smash them.
You don't want to smash anything else.
Yeah. So we don't want to keep this out too long because this is being now subjected to room temperature,
Right.
and so we want to protect the specimen. So if Kirsty gently puts it back in again.
So I guess when scientists do want to access a specimen from here, it's really important to try and be as efficient as possible,
Yes.
get it out quickly.
So you need to search on your database, you have to have the precise location, and you have to, it's very important you can come in and get it quickly, put it into your dry ice just like Aidan did outside, and take it to be packed up to send on to the researchers.
It does look very, it's quite hard to see anyway. It does all look quite mixed together in there. So, but I guess you kind of know exactly where you need
You
to go
do,
within each
yes,
tank.
you do have to know. So it's all, where Kirsty pulled that big tank out, there's a carousel that moves it round. So it occupies that whole space, and there's different regions that are, there's a postcode of address where that sample lives in there.
Okay, and so a quick final question for you. So these liquid nitrogen tanks, as we discussed, are much colder than the freezers that we looked at before, which I guess means it's slightly more effective preservation, or maybe
Yes.
for longer. Is it fair to say that maybe some of the more precious or valuable specimens get selected to put in here?
Definitely.
That
Yes,
must be
well,
difficult to
it's the best place to keep the sample stored for a long time, but it's the most expensive
Right.
still, really. So you know, the sample's got to earn its place here, and we do tend to put the most valuable samples here, and they can be valuable for different reasons. They can be valuable because the species is extinct or endangered, or it could be that they've been collected absolutely wonderfully from the field, and so, you know, the quality is absolutely perfect. So it would be a shame to put them in anything other than liquid nitrogen coming back from the field.
Well, it does sound like the place where David Attenborough should be putting his specimens.
He's been here and he's worn these gloves.
Nice. I hope they're feeding clean. Okay, we're going to wrap it up there, and thank you so much, Kirsty and Jackie, for showing us around this facility. We'll hand you back over to Alistair.
Excellent. Thank you very much, Camilla. And you can head in and get warm now after being in the chilly temperatures there. Great to see you in that space. And it's interesting as well that those little sort of drawers filled with specimens, these are the most valuable things that we might have in the museum. It's sort of strange to think that when we've got these wonderful fossils and skeletons and things out in the galleries.
Yeah, well, of course, it's a little bit subjective, but in a way, I think, you know, the point of 196 storage is that you don't get water crystals forming in the DNA on the sample, and that means that they can be preserved for so much longer. And in fact, we don't know how long they can be preserved for. But 196 is the gold standard. There are very, very few places that have that, and that's why it is reserved for the most precious samples at the moment.
Excellent. Now, just very quickly before we finish up, the Arks project, which Jackie mentioned in the link there, could you tell us just a little bit about what the sort of aim of that project is? Because this is not the only place where we're preserving these kind of samples, is it?
So I'm coordinating a project effectively to set up the UK's first coordinated zoological biobank, and that is about bringing together within a single database, and also in some very core collection facilities, including here at the NHM, genetic material from as many animal species as we can get from the planet. And the reason for that is that many of the populations and many of the species that we are working with, or have been working with in the last 20, 30 years, are extinct now. Some of the species are extinct, some of the subspecies are extinct, and if we don't do something very soon, we will lose all of that genetic material. And that genetic material is telling us about tens of million years of evolution. It's got new, different features, mutations in the DNA sequences, and that means that it's irretrievably lost when the species goes extinct. And that is why it's so important that we bring that together for research and for conservation. So, for example, with those bumblebees from 2013, if in 2050 the population is down to a very low number, and we've been able to measure the genomic diversity of individuals in 2013, we can see how much genetic diversity we've lost over that time, and we can make plans about how to put it back in again, whether that be using a different population or different individuals, or choosing the best individuals to breed from.
Fantastic work, and it's tremendous to hear about it. And unfortunately, we've run out of a bit of time today. I think we could probably chat for another hour about this sort of thing, but great to hear about the work you're doing, and all the best for it in the future.
Thank you very much.
And thank you very much, guys, as well, for coming along and sharing the night with us. And thank you very much, guys, over in the studio. Excellent. Thank you. Enjoy the rest of your evening. Thank you.
Good evening, everybody. Welcome. I'm Alison. I'm your host for the third of our talks this evening. And dare I say, welcome to European Researchers' Night, the biggest event on our public outreach calendar, as you may be aware. And this year, our theme of the evening is Planet 2.0. Now, we are basically focusing on the different ways that our scientific research is helping us better understand our planet and our impact on the planet, and dare I say, perhaps point to some ways we might have a bit more of a positive future for both ourselves and the planet Earth. And I am delighted to be joined by our third talk by Jon from our Department of Life Sciences. He's going to be talking to us about his particular field of expertise, the cephalopods. Now, I've got lots and lots of questions for Don tonight, but we would love to hear from you, our audience. This is very, very informal. We would definitely like to hear from you, so please, please don't be shy. If you've got any questions during our talk, raise your hand or call out. We'll bring a microphone over to you so that we can all hear, and we'll try to get through those questions. But let me introduce you to Jon, first of all. Thank you so much for coming to talk to us today, Jon.
No worries.
Tell us what you do here at the museum.
So I'm curator of molluscs here. So I should probably tell you what a curator does and what mollusc is. So I always tell children, I'm going to tell you the same because I can't think of anything better. Curators are like librarians, but instead of looking after books, I look after dead animals. I look after about 8 or 9 million dead, preserved mollusk specimens secreted around the building. And just like a librarian, we keep them in order, taxonomic order, like the giant tree of life, and we do research on them, and we help other people around the world to do research on them as well. So what's a mollusk? Mollusks are a really diverse group of animals. They include things like squid, snails, octopus, cuttlefish, seashells, bivalves, clams, mussels, and oysters. And although they all may look very different, we all think they all came from a common ancestor. So we group them together in a group called the mollusks.
And do they have any particular features in common that we can identify?
Yeah, so they are all bilaterally symmetrical, like us. So kind of you draw a line down the middle, same on both sides. They all, in their evolutionary history, show coiling. Some of them aren't coiled anymore, but their ancestors were. And they all have a shell, which you might thinking, you're talking about squid and octopus, where's their shell? But there is a shell. So there's mollusk. That's a seashell. I'm not talking about those. Those are gastropods. So that's a seashell. That's a cone shell. The shell is on the outside. It's obvious. The shell. Oh, I brought a shell. Shell. This is the internal shell of a cuttlefish, not just in budgie cages. So they have a shell internally, and they use them for buoyancy and for positioning themselves. And squid and octopus have internal shells as well. So they all have a shell made of calcium.
And it is the cephalopods, the squids and the octopus that we are focusing on today. So they're, again, quite a diverse group. So what comprises the cephalopods?
So the big ones are squid, octopus, cuttlefish, and then you've got some lovely weird groups like vampire squids, spirula, bobtail squids. There's always weird, wonderful little things. But yeah, mainly squid, octopus, and cuttlefish. They're the biggies.
Now, they have some amazing abilities, don't they? Not least their ability to change colour. Now, how do they manage to do that?
So the way they change colour is amazing. And you think of colour-changing animals, and people think of chameleons. And chameleons are rubbish at changing colour. I don't know why they think that. So for a chameleon to change colour, they need to think about it. They send chemicals around the body. It changes colour. It takes ages. Colour change in cephalopods is under nervous control. So they basically, you think blink. That's what they do. They think, change colour, and they change colour. So how do they do it? This is my patented cephalopod demonstration. So they're covered in cells called chromatophores, which are a bit like my glove. When they're deflated, they're mainly kind of a drab colour, but they inflate them under nervous control, and the pigment that's inside expands, creating a block of colour. And they do this all over their body, tens of thousands of them. So they inflate all the blue ones, they go blue. They inflate all the red ones, they go red. They've got mirrored cells, they've got iridescent cells, and just like your paint easel at school, you know, by mixing different colours, they can appear to show different colours in their environment.
And we've actually got a lovely bit of footage to show you. If we play that now, and Jon, if you can explain exactly what we're looking at.
Sure. So this is a section of squid tissue, and they pass an electrical current through it, and you can see that different electrical currents produce different cells to expand. So that's how the octopus does it. By sending different nerve signals through the body, it changes different pigmented cells to expand, stretching their pigment cells, and then they change colour. This isn't in real life. This is actually with a dead piece of squid tissue. In fact, if you eat squid, and they're quite a sustainable species to eat, if you run your finger down a squid's body and it still fires the chromatophores, it's super fresh. So you get food tips as well.
Wow. Try that when you get home. It is absolutely remarkable. So what is this particular skill useful for? How do they use this colour changing
So they
ability?
use it for loads of things. It's kind of the biggies. It's catching food, not being eaten, and finding a mate. What else is there in life? It's
So the camouflage is a particularly interesting ability they've got there. So we've got this fantastic example here. Look at that. So they're not just changing colour, are they?
No, they're changing the texture of their skin as well. And not only are they covered in chromatophores, they're covered in little structures called papillae, which are kind of like hair-like structures, and they can lie flat, giving them a flat view, or they can be pointed up into little points and everything in between. So they're changing colour and texture, which means they can perfectly blend in with their environment. And the thing that this is going to blow, I hope it blows your mind, it blows my mind. There's about 1,000 species of cephalopod, and apart from one species that we know of, they're all colourblind. So we don't really know how they match the colour that you see in the environment with the colour of their body. There's some theories to do with the way that the wavelength of light hits them, but I don't know enough about that to talk about it with any sense of understanding.
That is mind-blowing, isn't it? Absolutely crazy. So is anyone actually looking at that, researching into that?
Yeah, so I'm not very good at explaining this because it's not my field, but you know when you turn your camera on and the lens goes back and forward, it's focusing on light at different depths, and it kind of, that's how it works out different colours. And we think it does that by moving the lens in and out. It focuses on different colours and it can construct colour artificially. But there is one species that has 4 different pigment kind of rods and cones in the eye, so they can definitely see in colour.
Wow, it is amazing. Now, this camouflage is obviously excellent for avoiding being eaten, but there is some species that take it a little bit further, don't they? Can you tell us about this one here?
Yeah, this is brilliant. This is the mimic octopus. There's another species that does it called the Wunderpus, which is an awesomely named octopus. They both exist in the waters around Australia, Thailand, that kind of region. And they mimic shape and movement of more dangerous animals. So all octopuses are slightly venomous, but most of them are not that venomous. In fact, there's only one that can hurt humans. So it wants to basically be scarier than it actually is. So if you see on the left, these are all what we think. I mean, this is humans trying to understand behavior. We think that the octopus is trying to mimic the animals on the right. So at the top, you have the octopus in its natural state. Below that, you have a flatfish. And then below that you have a lionfish, and on the bottom you have a sea snake. And I mean, they're not amazing matches, but they're pretty good. You've got to remember that lots of things in the ocean don't have amazing sight, but not only are they matching the colour and the shape, but they're also matching the movement. So when it's pretending to be a snake, it kind of wiggles like a snake. When it's pretending to be a flatfish, it moves like a flatfish. And it's hoping, and all these other animals are far more dangerous, they're far more venomous than it is, so they're hoping to say, don't eat me, I'm scary.
It's pretty impressive, particularly the snake one.
The snake one's pretty awesome, especially because most of the body's buried, so it's burying in the sand, trying to be avoided.
It's ingenious. So avoiding being eaten is one use. Can they use their colour changing to actually catch prey?
Yes. So something that's amazing is the broad cuttlefish actually uses, and cuttlefishes are kind of the masters within the cephalopods. They, by strobing the chromatophores across, they kind of hypnotise their prey. I think, have we got a video?
We have. We've got a video that we can play. It's absolutely remarkable.
Yeah, so people don't really know how this evolved. One theory is that what they were initially doing was mimicking the light across the surface of the water, so that seems obvious. That's something in their natural environment. And they learned that they could use it to hypnotise their prey. So you can see this is a broad club cuttlefish going after I think it's a crab in this video, but they've seen it doing it to fish as well. And the crab just doesn't run away. It's kind of mesmerised. I'm pretty mesmerised. And then once it's kind of hooked, they shoot out their 2 long tentacles and catch the food. Pretty awesome.
Very, very impressive, isn't it? Do we have any questions from our audience so far for John? One second, I'll come over with the mic.
So for how long can those pigment cells stay expanded? Is it, we have 2 types of muscles in our body, one which needs constant stimulation to stay contracted and expands and needs energy.
It's those ones.
Yeah, it's those ones.
So they need nervous control to expand it, and then it just goes back again.
Okay.
So that's why when you buy a squid, it's normally white, or because all the pigments of the chromatophores have kind of closed up, concealing the pigment.
So can they actually get? I know it's a weird question, but can they actually get muscle pain from mimetizing too much?
That's a good question. I don't know. I don't know.
That's a very good question. You Fox-star scientist, that's brilliant. I like it when that happens.
I'm going home now. Can I have an easy one next?
Oh, we've got some questions here.
Does those pigment cells ever die?
So they do. So I brought some specimens out from the collection, and some of these are pretty old. The oldest one there, I think, is from 1831. And you can see they're pretty much all beige. So pigment doesn't stay in museum specimens when we preserve them. structural colour does. So that's why when you see things like butterflies in our collections or snakes in alcohol, they often look very beautiful because that's structural colour, but the pigmented colour fades over time, sadly, and everything goes beige. Which is why in old expeditions, they'd often take painters with them, artists, who would take beautiful colour drawings of these animals where they were fresh and alive, and now we use digital camera, which is good because I can't paint, so...
Do we have any more questions? I think we've got one right at the back there, keeping our mic runner on her toes, right at the back in the middle. It's just coming around.
Are the colours just in the visible spectrum, or are there any...
No, they are known to use colours outside the visible spectrum. So one interesting thing is that they possibly use colours outside our spectrum to, especially polarized light, to communicate within species, because then they can, especially in midwater squid, they can flash messages that can't be read by their predators. So, yeah, by using things outside the spectrum of most other animals, they can communicate in secret.
Ingenious. Any more questions before we move on? Okay. Now we've just mentioned communication there, so they obviously use to communicate within species. There are some species of cuttlefish that've got quite a sneaky way of using their colour changing, aren't there? Can you tell us about that?
Yeah, so actually is there a slightly different one first, just because that's a tricky one? No, not that one. Maybe it's that one. No, let me have a look. Oh, I can't see it. Oh, well, that'll do. The other one I'll do, sorry. So there's a species called the giant Australian cuttlefish. We have one here. They live in Australia, surprisingly, and they are the largest species of cuttlefish. Mantle length, which is the body of about 1.5 meters, about 2 meters when they're fully grown. And they live in harems. So lots of animals live in harems. And harems is where one, usually the male, gets very big and strong, fights all the other males, and has dominant mating rights over a pool of females. And lots of animals do this. You think of stags, mating, things like that. And that's fine if you're a big, strong male. You get to fight all the other males and you get to breed. But what happens if you are a small male giant Australian cuttlefish? Normally, you live on the edges of society and you don't pass on your genes. But what these do is they actually change their colour, they change their posture, and they change the way they swim, and they live as females within the community. So females are a slightly different colour. They have shorter tentacles and arms, so they pull them in slightly, and they swim differently. I don't know how female cuttlefish swims, but they swim slightly differently from the males. And they live as females within the community, and they get mated with by the male because they're tricking the male into thinking they're females. And when his back is turned, they change their coloration again into males, and they mate with the females behind his back. It's actually called sneaky mating. And it's been shown in lots of different species. It's been shown in fish, it's been shown in cephalopods, it's been shown in birds as well. This is incredibly successful technique if you're not big and strong. They've only studied it in aquarium studies where you can easily sample the next generation, but in aquarium studies they've shown that 30% of the next population are fathered by these smaller, sneaky males. So it's a really good way of doing it. And that's amazing, because if you think the amount of colour they have to do, what I love is this is in an aquarium, so this is a fake environment, obviously, and it's never been seen in the wild. There is a male there that is showing female colours on one side and male coloration on the other side. And you've got to think in an aquarium, you're in an artificial situation where everything's very close together. So the male might be really close, but he's still trying his best to be a male on one side and a female, so to mate on one side and not get beaten up on the other way. I can't pat my head and rub my tummy, at the same time. How do you maintain this? It's just amazing.
I do believe we might have a little bit of film footage that we can play that might actually show these guys in action.
Yeah, so you can see them mating. These are, like most cephalopods, they don't have an external penis, so they have to reach inside them, pull out a sperm and deliver it straight to the partner, and that's what they're doing here. But you can see the big dominant males fighting in order to have a place in the mating.
Sneaky mating, I love it. It's absolutely ingenious. Do we think that cephalopods are quite intelligent?
Yeah, so they've got about 500 million neurons. That's more than your cat, if you have a cat. It's about the same as your dog, if you have a dog. That's pretty intelligent. And the crazy thing is 2/3 of those neuron clusters are not actually in the brain. So invertebrates, which we're mainly used to when we think of animals, we have a centralized nervous system in the brain where most of it is. And you have the nerves and neurons across the body. But in cephalopods, they have these like little brains in each arm, and 2/3 of their brains, I think I can't really call them, but brainlets, are in these arms. And that means that each arm can act independently without going through the central nervous system so it can react quicker to the environment. They've got the largest brain-to-body size ratio of any invertebrate. They're the most intelligent invertebrate. They can solve mazes. They've got long-term memory. They've been shown to recognize human faces, which is quite remarkable, really. Often species don't sort of distinguish outside their species, but these have shown to know the difference between human species, let alone within their own species or related species. They can solve tasks, do food tasks. Yeah, amazing creatures.
And I'm sure most of you would have watched Blue Planet 2 when it was on television. There were some amazing behaviours observed there, wasn't there? Hiding, using shells, trying to suffocate a shark. Are we finding all sorts of new behaviours all the time?
Yeah, so two things. Blue Planet was amazing for so many reasons. But one of the things is it showed new behaviours never observed by scientists. One of them was tool use. So there's been some suggested tool use, because tool use is quite hard to prove. How do you know you're using it for a particular task and you're not just kind of picking up and playing with it? Play is also another thing. To play is linked with intelligence, but how do you know something's playing with it? Sorry, that's another topic. But they actually showed the veined octopus, again from around the waters around Indonesia, Australia, picking up shells, coconut shells, and holding them like a little shield around them. And then when something came, they'd actually climb inside the coconut halves, put them together like a portable shield. And the other thing that I thought was amazing was actually showed them cooperating with a completely different species, a group of fish, and the octopus and the group are working in tandem to try and catch food together, which is absolutely amazing.
It's remarkable. I think we've got a question here, do we? Have we got our microphone?
I forgot.
That's all right. No
I
problem.
just remembered. In Blue Planet 2, we've watched this film, and this fish was trying to open these shells when it had its home, and he used it took without hands. It took this shell of food inside. It wanted to eat whatever was inside, and it hit the shell on the rock, and it just smashed open. It just ate its.
So the thing is, fish are okay, but molluscs, cephalopods are even better than that. So lots of species of octopus do just like that. They pick up bivalves that are stuck together, and they smash them against the rocks. And they smash them to open them to get to the meat inside. Now, the difference between octopus and fish when they do it is octopus will try it one way, and if it's not working, they try a different angle, which fish have not been shown to do. So the octopus is showing a kind of learning. If you bash a certain species of mussel in a certain way, they won't open. You need to try the different end. And octopus have showed learning for opening food, so even better than fish.
Fish are pretty good. Octopus are better.
Maybe in the future they'll show the same things as octopus.
Exactly, and that's the lovely thing about Blue Planet. They're always learning new behaviours and things that we never saw.
And they might even discover new plants.
Possibly. Excellent question. Thank you. Have we got a question at the back there?
No? Oh.
Do any of them learn from other cephalopods, or does each one just need to learn all of it on their own? So do they pass on knowledge?
So there seems to be a bit of both. There's not been many studies, but there seems to be both. There seems to be some innate behaviour that seems to be passed on, but they also show learning as well. For example, in mimicry, you can put them on. Scientists put them on a chessboard. They're unlikely to have ever seen a chessboard before, and they make chessboard patterns. So obviously, camouflaging is not something. They're not just camouflaging the things they are in their natural environment. They're surveying their environment. But yeah, there's been quite a lot of research on that, and it seems to show both types of learning. But yeah, it's quite a new area of study.
Great question. Thank you. Any more before we move on? Yeah, we've got a question here. I'll come over with the microphone.
As a follow-up on the learning question, you showed a species which is capable of imitating bigger and more venomous predators, or anyway other animals. Is that behavior learned by observing this species, or is it innate?
I mean, we don't know. I mean, how do they know they're more dangerous? Is it learnt behavior from being in the community? But actually, most cephalopods are solitary. There are very few that live in big groups. I mean, that's one of the amazing things about science is we just don't know. And that's, one of the reasons we all who work here come to work, because we want to know these things. And because some of them are so hard to keep alive in the lab, we don't know their exact requirements, and many species don't perform very well, and they live in areas where it's very hard to film them. Yeah, we just don't know.
Now, we have in our collection, don't we, one very famous cephalopod. How rare, John, is it to have a giant squid?
Yeah, this is my biggest cephalopod. This is 8.62 meters long, called Arks, because that's short for the scientific name Architeuthis ducks. This is the world's largest, most complete giant squid. Few museums have got bigger bits, but they're not complete. This one was caught off the coast of the Falkland Islands in 2004. And so the fishermen sent it to me here at the museum. We put it on display. So 8.6 meters long. We think they get to about 13 meters in the case of the females, 10 meters for the case of the males. Have a worldwide distribution. You get them everywhere, just not in the polar regions. Occasionally they wash up in the UK. Washed up in Scarborough in 1936 and a few in Northern Ireland, Scotland more recently. But yeah, giant squid are real.
And quite elusive creatures as well. Is there quite a lot we still don't know about them?
Yeah, well, I mean, I started giving talks about giant squid in 2004, really, when we got ours. And people would ask me questions like, we don't really know. But actually, in the last 10 years, we've learned quite a lot. So we now know how many species there are using DNA tests on this and other more recent caught specimens. So we know there's only one worldwide globally distributed species of giant squid, Architeuthis ducks. Before that, it was postulated there was 7 to 13 sort of regional species. We have now observed them in the wild for the first time, first through photography and then video, so we know they're solitary hunters. They don't live in groups. We know they're active predators, actively hunting their prey. We don't think they come to the surface very often, but they migrate up and down to avoid things like sperm whales, which are their major predators. So we're learning more about them all the time. And, as I go on and give this, hopefully we'll learn more about them.
Absolutely. Remarkable to have it here at the museum. Very, very lucky. We also have quite a strange example here, don't we?
Yeah, I think this is my favourite
this
cephalopod.
Interesting cephalopod.
Everyone should have a favourite cephalopod. So this is brand new. This came to the museum. Which camera will you use? Should I hold it up or leave it there? That's what I'm saying. So this is maybe I'll hold it. So this is a vampire squid, which is confusing because it's not a squid. It's more closely related to octopus. The Latin name is Vampiroteuthis infernalis, which means the vampire squid from hell, which is pretty cool. These live in the kind of really dark areas, 2,000 plus meters down, sort of subtropical temperate waters all around the world. And they're not squid and they're not octopus. They have 8 arms. They don't have 2 tentacles, but they have these strange little filaments that no other group have, so they're all a little group of their own. They eat marine snow, which sounds cute, but basically that's all the stuff that dies at the surface, falls down as flakes, and that's what they feed on. They live in such an oxygen-poor zone that they can't really move very quickly, so they have a really unique way of dealing with predators. The first thing they do is they turn themselves inside out, called pineapple posture, and they expose the hairs and spines on the inside and this sharp beak. And if that doesn't put people off from eating them, they eject a glow-in-the-dark mucus. It's not ink. It comes from a completely different structure. They actually don't have an ink sac. And it's a bit like, I guess, someone turning on the lights while you're fast asleep. It brightens up a very dark environment, probably damaging the eyes of the creature that's only used to seeing in the gloom, and maybe blinding them and leaving this time to slowly plod away in the opposite direction. But this came in only the other day, so I have about 8 or 9 million specimens in the mollusk collection, and we always want new stuff. And this specimen came off a deep water trawl off the coast of Tristan da Cunha and just came to the museum last month. So, yeah.
Excellent. So it's an exclusive.
It's exclusive.
It's its first outing. Amazing. What kinds of research are we doing on our mollusks and our cephalopod collections?
One thing I'm involved at the moment is looking at isotope analysis. So you can look at some of the hard parts, like the beak. The beak is in the middle here. So all squid and octopus and cuttlefish have a beak. It's kind of the hard feeding structure. There's one there. So we're analysing these, looking at life history. So by looking at the different isotopes in the beaks, you can try and work out where it's been as it grew, which parts of the world they were found in. We can do some aging studies on them, trying to work out how long these live for. Most squid and octopus only live for a year or two. They spawn and then they die. But we think that vampire squid live for up to 20 years. They mate multiple times, laying eggs after each time, but we don't really know, so we're doing studies like that. Still trying to work out the tree of life of cephalopods. There's only 1,000 species known. There's probably many more undescribed, but we don't really know how they're related to each other. Looking at using preserved specimens to look at stomach contents. So for things we can't study in the wild, trying to look what's in their stomach now to see if we can work out what they eat, and then looking at them in other animals' stomach contents as well to try and work out where they fit into food webs and things like that.
If it's not an obvious question, why is it so important to understand the biodiversity of our oceans?
I mean, one reason is it's not. And I don't, Yeah, it is. But I don't think there's anything wrong with humans just wanting to know. Wanting to know how isn't a bad reason not to know. But there is good reasons to study. Firstly, is things like overfishing. So you take out some of the fast-watering, tasty fish, things like tuna, and you get squid and octopus that take their place as the primary predators in reason. Lots of people eat squid, so you're already increasing squid numbers by taking out primary predators. Then we start to overfish the octopus and the squid, and then what's going to take their place? You get jellyfish. We'll all be eating jellyfish burgers in 100 years' time. So understanding the delicate balance of the environment, knowing the interconnected webs, knowing what's out there, knowing what's, and on a fisheries point of view, knowing what's sustainable, what breeds quickly, what should, because we are going to have to feed our planet using fish, what should we be taking, what things grow quickly. Octopus grow very, very quickly, very, very shortly. Maybe that's something that we could look at in the future. What affects, so acidification, as our temperature changes, the balance of calcium in the water changes through acidification, and all molluscs need to produce a shell, and if you change the acidity, the shells are made of calcium, they could dissolve, they could become unsustainable. Also, squid and octopus have hemocyanin, not hemoglobin, so they use copper to bind their oxygen to their blood, not iron like we do, hemoglobin. And by changing the temperatures of the water, you change the efficiency of them being able to bind oxygen into their blood. They don't actually bond as well as hemoglobin does. So again, you're going to change these species that possibly can't survive in areas. They're going to be moving to new areas, upsetting ecosystems. So there's so many reasons, as well as it not mattering.
Yeah, even the very, very deep sea creatures, we could still find a way to have an impact on those. So yeah.
And also, chemicals, we don't know. So you might see the vampire squid's gone purple. We don't know what pigment causes that. So, whenever you think chemicals, there might be some industrial use and medical use, the venom inside them, lots of uses.
Case in point.
Yes.
Blue-ringed octopus here.
So I said everything goes beige, and sadly all our blue-ringed octopuses have gone beige, so they're no longer bright yellow and blue. But this is a blue-ringed octopus. This is about as big as they get. Made famous in James Bond Octopussy, if you've seen that. They are the world's most venomous octopus. So all octopus are slightly venomous, which means that when they bite you, they inject a toxin in, and they don't produce the toxin. It's done by some bacteria in their salivary glands, and when they bite you, the saliva goes into the wound, and it's used mainly to slow down predators. There's some meat tenderizing factors in there, but also slightly affects the nervous system, slowing them down. But these are so venomous that they can kill a human with one bite. They live in Australia, like everything poisonous, also Thailand and Vietnam. Apparently, if you get bitten by one and you get enough venom in, you've got about half an hour. You have to keep either get anti-toxin or you need someone to give you artificial CPR to keep you alive. It takes you half an hour to process the toxin. But they're bright yellow and blue, and they're saying, do not pick me up. So actually, when people get bitten, it's usually because they see a yellow and blue octopus and pick it up and go, oh, look at this. But that's what they're using colour to say, do not pick me up. It's just like wasp stripes. It's like, venomous snakes. They are making themselves very conspicuous because they are deadly.
But lots of venoms have applications, don't they, for medicines and that type of thing?
Certainly.
So, yeah.
I don't think there's any been taken out of this, but I was sure people will be looking at it in the future.
Do we have any final questions before we wrap up, guys? Oh, yeah, we've got a hand up here. If we could get the mic over.
How do you catch one of those venomous octopuses?
Carefully. I've never collected in Australia. I don't think I want to. It's too dangerous. Some kind of net or a glove, I guess. But yeah, I know that people who collect. So this is Dosidiscus gigas. This is a really aggressive squid from Chile, coast of Chile, and they hunt in packs of up to 1,000. Very, very dangerous. They've been known to kill people, and people that go to collect them actually wear diving chainmail because they're so vicious. They don't want to be bitten. So yeah, something like that, I would imagine.
Have you ever filmed a giant squid taking down a ship into the water?
So people laugh, and we don't think they get quite as big as in the old paintings. They're not going to drag down a boat. But I have personally seen the hull of a US submarine that has marks on the front that look like squid suckers. And I don't think it was trying to take down the submarine, but they are intelligent, inquisitive
Maybe
animals.
defending.
I think just investigating. They're intelligent animals, and when something new, like a submarine, comes into their environment, they want to, they want to investigate. They want to play. And there's marks on the front of a hull of a submarine that I am
Like a
swear.
worm and a fly. They didn't know what's each other.
Yes.
Excellent. Thank you. Unfortunately, guys, we are out of time, I'm afraid. But don't worry, Jon is out on the galleries after this talk. He's in our Variety of Life zone, aren't you, in Dinosaur Way? So if you want to ask Jon any further questions, do go and chat to him there. He'd be more than happy to talk to you guys.
And you can see some slugs. Everyone likes slugs.
Yes, Jon likes slugs and snails as well, so he's got quite a few of those on the station. Thank you so much for coming down to our talk today. You might have noticed on your seat there are some evaluation cards. If you are happy to fill those in for us and pop them in the box over there, that would be absolutely fantastic. We hope you enjoyed our talk. We hope you enjoy the rest of our night. Thank you very much for coming. Can you join me in thanking Jon?